Hi, welcome to my YouTube channel. Today we'll be talking about the folding of proteins, a continuation from my previous video. The folding of a protein chain can be constructed by many different setbacks of weak non-covalent bonds that form between one part of the chain and another. So these are going to involve the polypeptide backbone as well as atoms in the amino acid side chains. So the weak bonds are of three types. We have hydrogen bonds, ionic bond, and vanderwaals interactions. Individual non-covalent bonds can be 30 to 300 times weaker than the typical covalent bonds that create biological molecules. But many weak bonds can act in parallel to hold two regions of a polypeptide chain tightly together. So the stability of each folded shape is therefore determined by the combined strength of large numbers of such non-covalent bonds. So we have three types of non-covalent bonds that help proteins to fold. Although a single one can be quite weak, many of them are often formed together to create a strong bonding arrangement. So, how a protein folds into a compact conformation, we are going to have the polar amino acid side chains. They tend to gather on the outside of the protein where they can interact with water. So the nonpolar amino acid chains are buried on the inside to form a tightly packed hydrophobic core of atoms that are hidden from water. So hydrogen bonds in a protein molecule. So we are going to have large number of hydrogen bonds formed between adjacent regions of the folded polypeptide chain and help stabilize its three-dimensional shape. As a result of all these interactions, each type of protein has a particular three-dimensional structure which is determined by the order of the amino acid in its chain. So the final folding structure conformation adopted by any polypeptide chain is the one which has a minimized free energy. So a protein can be denatured and folded by treatment with certain solvents which are going to disrupt these non-covalent interactions holding together the folded chain. So this treatment converts the protein to a flexible polypeptide chain that has lost its natural shape. So when the denaturing solvent is removed, the protein often refolds spontaneously or renatured into its original conformation. And this indicates all the information needed for specifying the 3D or three-dimensional shape of a protein which is contained in its amino acid sequence. So each protein normally folds up into a single stable conformation. However, this conformation often changes slightly when protein interacts with other molecules in the cell. So this change in shape is very important to the function of protein. Okay, so even though a protein chain can fold into its correct conformation, without outside help, protein folding in a living cell is often assisted by special proteins called molecular chaperones. So these proteins bind to partly folded polypeptide chains and help them progress along the most energetic, favorable folding pathway. So chaperones are very important because the they are going to help these proteins to fold. Okay, so they are important also in crowded conditions of the cytoplasm, since also they are going to prevent exposed hydrophobic regions in newly synthesized protein chains from associating with other to form protein aggregates. So the final three-dimensional shape of the protein is still specified by its amino acid sequence. Chaperones simply just make the folding process more reliable. Thank you.